Nucleotides and nucleic acids are among the most commonly recognized biomolecules in popular media. This, of course, is because of the importance of DNA as a carrier for our genetic information. Right? So DNA is commonly seen in things ranging from science programs to forensic science programs or criminal drama shows like that. Now, of course, from a biochemical perspective, DNA is a very important biomolecule because of that genetic carrier role. However, there are many other key functions of our nucleotides and nucleic acids. It's very important to take a close look at these and, again, learn how their structure relates to that function. Right? So we see nucleic acids involved in transmission of genetic information, carrying that from our nucleus to our cytosol and to our ribosomes. We also see it involved in processing genetic information and even directly involved in protein synthesis. These, of course, are some of the more common and popular roles of our nucleic acids, but there are also a wide range of other roles, even for nucleotides in the monomer form through our energy from metabolism. So that's our ATP involved in different signaling processes or as enzyme cofactors as well. So again, as always, in order to understand all these different functions of our nucleotide and nucleic acid class of biomolecules, we're going to start by looking at the basic structure of our monomers, and then we'll see how those are linked together into more complex polymers in order to carry out all of these various complex roles. One of the first things that we see as we look at the structure of our nucleic acids is that they actually have a backbone that's formed from our carbohydrates or sugars. So the aldopentose D-ribose forms the basis for all of our nucleic acid monomers, as well as the polymers that are formed from those monomers. We have either ribose, which we use in RNA, so it's a ribose nucleic acid, and we have our deoxyribose that we use in DNA. And that's where, of course, we get the deoxyribonucleic acid, which gives us our DNA acronym. As we mentioned when we were looking at our sugar molecules, the only key difference here between our ribose and deoxyribose is the presence or absence of this hydroxy group on our second carbon here in the ring. So the name actually, again, tells us exactly what that change in structure is. We've removed our oxygen here from our ribose structure. So while our sugar forms the backbone of our nucleic acid structure, we'll have several other key components attached to that sugar in order to give us the unique properties of our nucleic acids. So in order to form the base monomer of our nucleic acids, which we call a nucleoside, we're going to take our pentose sugar and attach a nitrogenous base on to the first carbon of our sugar. So you can see the exact identity of the base will vary, but it's always linked onto our sugar molecule to create our nucleoside. The common bases that are attached to our sugar are pyrimidine bases and purine bases based on the structure of the ring that's attached. Right? So notice we have a purine base here with that double ring structure and a pyrimidine base over here with that single ring structure. We will see different varieties of both pyrimidine and purine bases. One of the things that you should notice here is we have now a cyclic system that's attached to another cyclic system. So we'll have one numbering system for our base, and we have a separate numbering system for our sugar ring. So you might have noticed on the previous slide that each of our carbons in our sugar were labeled with a little apostrophe to indicate that we're talking about the sugar ring and not the ring of our base here that's attached to the sugar. For most of our nucleic acids, we will also attach a phosphate to our sugar ring in addition to that base. So this forms what we call a nucleotide. So the nucleoside is our base monomer with only our sugar and our nitrogenous base. And our nucleotide is when we include the phosphate on there in addition to our sugar and our base. So phosphate, of course, This is our inorganic phosphate with our oxygens attached to our phosphorus, and so it forms a phosphate ester that attaches to the alcohol on the 5th carbon of our sugar. Right, so we had our alcohol here, which now is linked into this phosphate ester with our phosphate group. Some of the key features to notice here with our phosphate group, so of course, as always, our phosphate group is going to be negatively charged. So we'll add on some negative charge here that our DNA can use for forming interactions with other biomolecules. It's also generally going to be attached at the 5' position of our sugar ring. Again, as I mentioned, it's attached to our 5th carbon. It could technically be attached to any of our hydroxy groups. Right? All we need is that alcohol to react with our phosphate and form that phosphate ester. But the 5' position is the normal position for that phosphate to be attached. We will see cases where it's attached in other locations, but those will be more special cases. Here you can see some of those different varieties for where we could attach the phosphate. So again, we can indicate where we attach it just with a number and our little apostrophe to indicate the carbon position of our sugar ring. So we can have a 5' monophosphate where it's attached to its 5th carbon, a 2', a 3', or even we can attach it to both at once. So we can form 2 different phosphate esters and have a 2', 3' cyclic monophosphate. While the phosphate of our nucleotide is very important for its negative charge and capability then for ionic interactions, the key component that makes our nucleotides unique is our nitrogenous base. So this is where we'll see most of our variation between our different nucleotide monomers. So again, we'll have either pyrimidine or purine derivatives, and we'll attack. While the phosphate of our nucleotide is very important for interacting with other molecules using that negative charge, it's really the nitrogenous base that makes our nucleotides unique from each other as well as from other biomolecules. Right? So every nucleotide will have a phosphate group attached, normally at that 5' position, but our base will be different for each of our unique nucleotide monomers. Now, as I mentioned, they will either have that pyrimidine 6-membered ring structure, or we'll have our purine double ring structure with our 5-membered ring that's fused to our 6-membered ring. Each of our rings, of course, contain nitrogen, so these are our nitrogen-containing heteroaromatic rings. So notice the presence of our alternating double bonds. Of course, nitrogen can still be in aromatic rings, just like our carbons can. So these are, in fact, both aromatic ring structures. Even our little 5-membered ring over here on the right, our nitrogen here can share its lone pairs in with the ring to keep this an aromatic system as well. Because of that aromatic structure, that also means that these are always going to be nearly planar structures. Right? Aromatic rings have to be planar in order to allow for the overlap between all of our adjacent p orbitals. Unlike our proteins and our carbohydrates, there are relatively few varieties of monomers for our nucleic acids. So we have 4 main nucleic acids that we find in DNA: cytosine, adenine, guanine, and thymine. And then we have one additional nucleic acid that's only found in RNA, and that is our uracil. So DNA and arginine both share cytosine, adenine, and guanine, and then we have one nucleic acid that's unique for each of our main nucleic acid polymers. Now, I should note that these are just the most common varieties. There are some other forms of monomers that we will see occasionally, but they're much less common. So it's a similar case as with our proteins, where you have the 20 main amino acids, and then there are some other minor amino acids that you might see used occasionally in specific scenarios. All of these different bases are very good at forming hydrogen bonds with multiple different hydrogen bond donors and acceptors. They also all are neutral molecules at pH 7, So we don't have any charges here that we'll be using to interact between these bases. It's really just going to be focused on forming those strong hydrogen bonds between our bases. Here we see the structure of our first two bases, which are both formed from the basic structure of our purine. So we have adenine here on the left and guanine on the right. They both have that double ring structure, but then with a few additional functional groups added. So you can note we have our amine groups, which I've circled here. So our amine groups make great hydrogen bond donors, which again, as I mentioned, our bases are going to be filled with different hydrogen bond donors and acceptors that we'll use to form hydrogen bonds between our bases. Our guanine also has a great hydrogen bond acceptor added in the form of our carbonyl. So I'm going to draw a square around this hydrogen bond acceptor. So we have our acceptor here for guanine, and we have these hydrogen bond donors on both adenine and guanine from our amine groups. Now, of course, it's important to note that our purine ring itself also has several hydrogen bond donors and acceptors. So notice on guanine, this nitrogen here can act as a donor. On our adenine, our hydrogen is removed to make room for a double bond, and so this nitrogen can only act as a hydrogen bond acceptor. We also, of course, have several other nitrogens on our ring structure that could also be potentially used as either donors or acceptors. So here we have a couple more hydrogen bond acceptors. And of course, with our hydrogen attached to this nitrogen here, it could act as a hydrogen bond donor. And we see the same thing over here on our guanine. There are no further differences between our two structures beyond those three changes on the left side of our structure. We have three different bases that will be formed from our pyridine base ring structure. So we have cytosine on the left, thymine in the middle, which of course is only found in DNA, and our uracil on the right, which is only found in RNA. So they all have the same basic six-membered ring, but again, with different added functional groups on to change our hydrogen bond donor or hydrogen bond acceptor capabilities. Again, I'll circle our hydrogen bond donors. So we have an amine added to cytosine to make a great hydrogen bond donor. We also have hydrogen bond donors on our pyrimidine ring structure itself. So we have one each on the left side for uracil and thymine, and we have one on the bottom from all three structures. When we look for hydrogen bond acceptors, so we see, again, mostly carbonyls being added on to create hydrogen bond acceptors, although we do also have a nitrogen on our cytosine that can act as a hydrogen bond acceptor, but not a donor, because it has that double bond instead of the hydrogen. Now, I should note that our hydrogen bond donors do have the potential to accept hydrogen bonds as well, because they would have an extra lone pair there as well. But I'm emphasizing their hydrogen bond donor role because we have a limited hydrogen bond donor capabilities on our ring structures. Our thymine and uracil both have two added carbonyl functional groups to act as hydrogen bond acceptors. And you might notice here, as you look at thymine and uracil, their structures are very similar. In fact, the only difference here is in our methyl group over here on the right side of thymine. So we have a methyl group on thymine and no methyl group on uracil. So as you should have noticed, each of our different bases have a different name based on their structure. So we have adenine, guanine, cytosine, thymine, and uracil. Of course, you might also recognize their single letter codes that are commonly used as well, right? So these are the famous A, G, C, and T, with that added U for our RNA base. Now, these are specifically the name of just our nitrogenous base. Again, remember, these are normally going to be linked to a sugar to form our nucleoside. When we make our nucleoside, we change our ending to a sine ending to indicate that we're now bound to our sugar here. So adenine becomes adenosine or deoxyadenosine if we're binding to deoxyribose. Guanine becomes guanosine, and so on. We do have an exception here for our pyrimidines. So they actually take a DINE ending for a nucleoside, and we get cytidine, thymidine, and uridine. For our nucleotides, we've now added on our phosphate. So in the first case, we added on our sugar. In the second case, we added on our phosphate. The phosphate, of course, introduces a negative charge, and so we use an ATE ending to indicate the presence of that negative charge. So now it becomes adenylate, guanylate, cytidylate, thymidylate, and uridylate. Again, using that HE ending that we commonly see with things like phosphates and carboxylates, all using that HEE ending to indicate that negatively charged carboxylate or phosphate group. So the most common form of our nucleotides then is shown here. So here are the 4 nucleotides for our DNA. You can notice our full names here with that adenylate ending and our deoxy at the beginning. You'll also often see the position of our phosphate noted. So this is a 5' monophosphate because we have a single phosphate attached. You'll notice that there are several different symbols we can use to represent our nucleotide as well. So we can just use A to represent our deoxyadenylate here, or we can use a DA to specifically indicate that it is the deoxy form as compared to the non-deoxy form that you'd find in RNA. Or we can specify that it's DAMP. MP would stand for monophosphate, indicating that we have only one phosphate attached. Right, so that would be as opposed to ATP, which would be adenosine triphosphate, where we have additional phosphates attached on here. Our other nucleotides follow the same basic format. Again, we have 3 different possible symbols that all could be used. G as the most generic symbol, DG getting slightly more specific, DGMP even more specific. For all 4 of these nucleotides, it is important that you know the structures, names, and symbols here so that you can easily recognize them in different biochemical scenarios. Now, of course, for our RNA, we have essentially the same 4 structures with just our few small differences. So we have our OH here for our ribose sugar instead of the deoxyribose sugar. We also have our uracil or uridine here instead of the thymine or thymidine that we have for our DNA. We also use the same key symbols here. A, G, and C will still represent our adenine, guanine, and cytidine nucleotides. We just don't have the DA or DG form since these obviously are not the deoxyribose sugars. One of the interesting things that we see with our nucleic acids is that our individual monomers are actually formed from those 3 smaller key units of our phosphate, our sugar, and our base. So as we saw with our phosphate, one of the ways that we can modify or change our individual monomers is by changing how our different 3 units are connected together. So with our base, we're going to connect it to our sugar using a glycosidic bond that's somewhat similar to the bonds that we use to attach our sugars together. But we call this a N-glycosidic bond because it's forming from a nitrogen to the anomeric carbon of our sugar. So it's actually quite similar to the N-glycosidic bond that we saw between sugars and proteins when we had things like hemoglobin getting glycated or different forms of glycoconjugates where we're linking those carbohydrates to our protein molecules. Now, specifically for our base, we're always going to link our N-glycosidic bond in this beta configuration, right? So beta, of course, is referring to the anomeric form. So beta would be cis with respect to the terminal carbon of our sugar ring. Using the nomenclature for our glycosidic bonds, we would write this as beta 1, the first carbon in the beta orientation from our sugar, to either N1, the nitrogen at position 1 of our pyrimidine ring, or N9, the nitrogen at position 9 in our purine ring. So we change up kind of where that attachment point is, depending on which ring structure we have. This bond is very stable toward hydrolysis, so we won't commonly see our bases popping off, but under more acidic conditions, we can break that bond, which would end up degrading our nucleic acid. So here you can see the structure of that beta N-glycosidic bond again. So notice, of course, how our bond is going up from our anomeric carbon, which of course puts it on the same side of the ring from our terminal CH2OH. Our N-glycosidic bond, of course, can freely rotate. As we've seen with our carbohydrates, right, it could actually rotate and form different conformations to make different shapes for our larger sugar polymers. In the case of our nucleic acids, we'll be concerned with two main conformations of our N-glycosidic bond. So either syn, where it's kind of pointing back in the same direction as our sugar, so it ends up kind of sitting right over our sugar ring here, or anti, where it's pointing out in the opposite direction away from our sugar ring. So notice we have our adenosine and cytidine here pointing out in that anti-conformation versus the adenosine here pointing backwards in that syn conformation, kind of hovering over our ring. So this would correspond to a 0-degree angle of our N-glycosidic bond for the syn conformation, or a 180-degree angle for our anti-conformation. We commonly tend to see the anti-conformation in our normal DNA structure, but there are some scenarios where that syn conformation will be useful as well. So the advantage of our anti-conformation is it kind of points those key hydrogen bond donors and acceptors out away from our sugar ring, where then we'll be able to bond with other hydrogen bond donors and acceptors. In addition to rearranging our beta N-glycosidic bond into different conformations, we can also rearrange our sugar ring itself. Now, of course, as you should remember, our sugar rings are all formed from this reaction of our original aldehyde or ketone, where we cyclize it through the formation of our hemiacetal or hemiketal in the case of our ketoses. Now, once we have this ring structure, so it can bend itself into different conformations. If you remember, of course, we have our different chair or boat conformations, which we talked about in depth when we looked at our sugar chemistry. For our 5-member ring structure here, we end up with this kind of puckering where we can kind of bend up one end of our sugar at a time. So we refer to these as either endo or exo. So you might remember that endo is a term used for rings when they're forming kind of this cis conformation. So notice how my ring is kind of bending up in the same direction as my 5th carbon and my base. Or we have this exo position where we're kind of trans with respect to those other groups attached onto our ring. So it's going to be bending down, whereas our base and our terminal carbon are both pointing up. Both our 2nd and our 3rd carbon can be puckered into this exo or endo conformation. So we have a little bit of flexibility there between both of those groups to kind of move around into different conformations. As we saw with our proteins and our sugars, just because we can bend our structure into different shapes doesn't mean that we will commonly do that in our larger polymer structures, right? So as we bend into certain shapes, we'll have steric hindrance between our groups that will prevent those specific angles. So the puckering and the different conformations of our ring will affect the conformations that we get with our beta N-glycosidic bond, and will also affect the conformations of our bonds here connecting us to our phosphate groups as well. While our base here on our nucleic acid might have multiple different conformations, it's important to note that our base only ever has that one attachment point to our anomeric carbon, since we have to form that glycosidic bond between our anomer and our amine. In contrast, as we saw with our phosphate, we can attach it in multiple positions based on where we have our hydroxyl groups on our sugar ring. So even if we have a DNA without our hydroxy group on the second carbon, we still have at least our 3' position and our 5' position, where we can react that phosphate with our alcohol in order to form that phosphate ester. These multiple reactive sites for our phosphates enable us to form our polynucleotides. So as we go to link our monomers together, we're going to use a single phosphate that will be linked to the 5' OH of one of our sugars to the 3' OH of our next sugar to form a phosphodiester linkage. So these phosphodiester linkages will be repeated in order to link many different nucleotides together and create our larger polynucleotide polymers. So we'll take a closer look at this polynucleotide structure in our next video.